Yongshen. We are very tight on time right now. So for those, I know that those people who are going to leave at 4, please, maybe Yongshen, you can shuffle, and make sure people are okay with their time. And so for the next few speakers, please do keep, to the time. I believe you were given, what is it, 6 minutes, 5 minutes?
Minute.
6 minutes.
6 minutes per person. So maybe I'll ask Yongshen, you guys keep the watch and then tell them that it's 6 minutes and we have to cut.
Oh my God, 6 minutes?
Yes.
Okay.
Surprise!
Thanks.
Okay, go ahead.
Okay. Good afternoon, everyone. Today we are going to present our project just like this. Brain connectivity during observed fear anticipation and loneliness reduction after meditation training. And we are Jenna Cho and Cha Ho from GIBMS of National Taiwan University. Both of us are in the lab team of Dr. Zhen Minzong. This is Cha Ho speaking for this pitch presentation. Before we begin, I want to ask a simple question. Do you feel lonely? So, previous research suggests that loneliness may be related to lower empathy. In simple terms, when people feel lonely, they may report that they feel less connected to others emotionally. Like Squidward in Chinese is Zhang Yu Ge, is probably the perfect example here. Very lonely, not very empathic, and clear, not interested in social bonding, just like a boring adult, just like us. But behind this joke, there is a serious scientific question. If loneliness is associated with a lower empathy, can we reduce loneliness and improve social connection? One possible way to reduce loneliness is meditation. In particular, loving kindness meditation, or LKM, is designed to increase warm and positive feeling toward oneself and others. So, like just previous paper we focus on is titled Lonelier. People feel less empathic despite intact neural empathy responses after meditation training. The study examined whether meditation training can reduce loneliness and increase empathy. It also used functional MRI to test whether loneliness is related to neural responses during an empathy-related task. So this is the flow chart here. They included 108 participants in the study and assigned them to either love kindness meditation or progressive muscle relaxation for 4 weeks intervention. After 4 weeks of training, participants completed another survey and underwent a functional MRI scan. Later, they also completed a follow-up survey. So there are 2 kinds of survey here. During the functional MRI test, participants experienced 2 types of situations. The first one is they directly experience painful or non-painful stimulation. And second, they observed another person receiving painful or non-painful stimulation. This design allowed the researchers to compare direct pain experience with observed pain or fear anticipation. The key idea is that observing another person's pain can activate empathy-related brain systems. So the main finding was interesting. Both meditation and progressive muscle relaxation reduce loneliness. However, the reduction in loneliness did not clearly lead to an increasing trait empathy. The brain imaging results also suggested that loneliness was not strongly associated with direct difference in neural empathic responses. So lonely people reported lower empathy, but their neural empathy responses seems relatively intact. So the original study concluded that both intervention reduced loneliness but not trait empathy. This means that the original hypothesis was not fully supported. Also, there was no strong evidence that loneliness changed basic neural empathic responding. This creates a new question for our project. Maybe there is some difference. Maybe the story is not finished here. Instead of only asking whether meditation changes empathy, we want to ask a broader question. Could meditation affect the social cognitive function? Social cognitive include how we understand other thoughts, emotion, intervention, and social signals. So our idea is that the loneliness reduction may involve not only emotional empathy, but also communication between emotional and social cognitive brain networks. So is loneliness reduction after meditation training associated with changes in brain connectivity during observed fear anticipation? This is important because loneliness may not damage empathy itself. Instead, loneliness may change how emotion signals are integrated with social understanding. So connectivity analysis may reveal something that standard activation analysis cannot show. So the knowledge gap here is that prior work mainly focused on regional activation or pattern similarity. Let's know about text-based functional connectivity during observed fear anticipation. Our aim is to examination whether loneliness reduction after meditation training is associated with a connectivity between affective empathy regions and mentalized regions. Our hypothesis is that participants with greater loneliness reduction will show stronger connectivity between the anterior insula and regions such as TPJ or MPFC. This association may be stronger in the loving kindness meditation group than in the relaxation group. We will use the open neural datasets from the original study. These datasets include functional MRI data from meditation training participants, as well as behavioral and questionnaire data. The data are already organized in BIDS, compatible structure, which is useful for reproducible neuroimaging analysis. This makes it a good data set for a student project. Our conceptual model starts with intervention LKM and PMR retraining. The intervention may reduce loneliness. Then loneliness reduction may be related to change in affective empathetic networks, such as the anterior insula and ADACC. Then this region may then interact with a mentalizing or social connectivity network, including TBJ, MPFC, STS, and PCC. Finally, this network living level change may relate to subject empathy or social connections. So this is our analysis methods here, and we don't have enough time. So thank you. Thank you. Thank you all.
Any questions? So is your theory is that
Is it too fast? Oh, sorry.
no, this is great. So I have one question. So is the idea here that loneliness doesn't affect empathy, but it affects the interpretation of the
Yeah.
signals?
Yeah.
That's your idea.
Yeah, that's my
Okay.
our ideas.
Okay, great. And the point is that you think that that can be validated with that data set.
Yeah.
That didn't show any difference. It's just that you're going to analyze it differently.
Yeah.
Got it.
Yeah.
Okay. Cool with me.
So the point is to survey the connectivity here,
Okay.
not just a regional.
The whole brain,
region.
the whole region. Okay.
Yeah.
Got it.
Yeah.
Okay. Any questions from others? No. No. If not, let's go on quickly to the next person because we
you.
keep catch with the time. Who's next? Great job. Just share a screen, let everyone know who you are, and you can take it away. 6 minutes. Hey, who's timing? Oh, yeah.
Hello. Okay. Hello, everyone. I'm Patrick. Oh, I'm Ding Yichen, and my partner is Chen Yuling, and we're from NTU Taiwan and Department of Psychology. Today, our project is about affordance and motor-related representation in the dorsal pathway. Specifically, we want to understand whether action-related information can be extracted during passive object viewing conditions. So before, so Before digging into our research question, let's start from some daily example of what is affordance. Different door handles naturally guide different actions, such as pushing or pulling. And also, I think most of us will tend to use spoon like the way on the left-hand side rather than the right-hand side. And also you can turn on the light with different type of switches, even if you never use this kind of switches. So this suggests that objects themselves can evoke specific action possibilities. So we want to know whether there is a neural representation that can encode different objects and encode their affordance rather than purely visual features. So before addressing this question, let's go to the definition of the affordance given by Gibson in 1979. And affordance are the properties that environments or objects offer to humans or animals. So in other words, affordance refers to the possible actions of an object depending on the perceiver's ability, intentions, and goals. Also, affordance can influence our perception and action by integrating different visual informations. Okay. So we want to know whether there has been some neural correlate of affordance. In a macaque research, researchers found that neuron in V6A can process affordance information rather than just purely basic visual features. So you guys can look on the figure that these neurons activity patterns are much more similar with the handled orientation rather than the thickness of the object. And also there has a lot of different studies reported that affordance or tool-related responses across broad dorsal pathway networks. So our researcher question is, but however, most of the previous study focuses on the action planning and action execution, and less lack of the study which focusing on whether the object representation can encode affordance or encode motor related features. So we want to know whether passive viewing of objects can really evoke some motor related representation in the dorsal cortex or in the motor related area. So to answer this question, we have three hypotheses. The first is in the dorsal and motor related regions, objects are organized based on affordance structures. And the second hypothesis is about objects with similar affordance should evoke similar neural representation, even if their visual features is different. And the third is objects that are visually similar may still evoke different motor representation. And we will test this hypothesis by analysis the neural activities in these ROIs and using things that are said. Okay.
So why do we choose things as our data set? Things is one of the biggest image database in the world, which contains more than 1,800 object concepts with 12 or more object images per concept. They recruit participants to watch those pictures and record their EEG, MEG, and fMRI data. For our project, we decide to analyze fMRI data to validate our hypothesis. The reason we choose things is that it collects brain imaging data of simply viewing images rather than responding to any other task. So we will compute a dissimilarity score for every pair of objects. This gives us RDM, represent each cell encodes how different the brain's response to one object to another. So the main tools or methods we will use is RSA and RDM in here. To answer our first question, you can see all of the pictures are the same concept of handle. but with different shapes, which lead to different affordance. So we expect to see the beautiful result like this picture we produce. Lever and knobs are quite different from one another. Okay. You can see a pitchfork, dining fork, spoon, and a pen here. These objects differ in their visual appearance and their affordances. The RDM wood captures the representational distance between all pairs. This allows us to see whether objects that look visually distinct are represent as similar, or if objects in the same category diverge based on their functions. So the third hypothesis, here we will compute RDM from two very different brain regions using objects like keyboards, calculators, and chessboards. If our hypotheses are correct, these two metrics should look quite different from each other. The motor areas should organize objects by their affordance structure, while V1 and the ventral stream should organize them by visual similarity. Okay, so thanks for your listening.
Okay, very, very good. Thank you. Awesome. So first, it's not going to be 这么漂亮. So any questions from people? Yeah, go ahead. You want the mic? You pass the mic. Thank you.
All right, so basically I was thinking about even if you find the significant fossil in the affordance, how do you know that the significance is really comes from the affordance, not from similar visual feature or basic concept like I can grab this thing? You understand?
I think the concept that I can grab these things is the definition of affordance, and how we can rule out that this isn't represent the vision features is that the ROI of our research is focusing on more motor related regions, and we want to compare it with some vision related visions like ventral pathway and dorsal pathway. I think dorsal pathways may encode more motor related features than ventral pathways, because object recognition is ventral pathways is responsible for object recognitions.
So are you going to have, they have brain data? You said how many people?
3 people.
3 people looking at a lot of all these pictures.
Yes, and all our pictures.
And then you're going to, for these 3 people, define their dorsal or ventral region. Is that how you're doing? Which template are you going to use to define these regions?
We haven't had any idea to really define the ROI.
So that will be a very critical step, because if you choose the wrong ROI, it seems that the ROI is very important. Yeah. If you choose the wrong ROI, you might get not valid results. So that would be a very critical thing. You might want to ask the TAs about templates, or if you don't have some candidates already.
Okay.
Yeah. Any other?
Just
online. Yep.
Yeah. Very interesting project, and I actually believe that there might have been some work looking at this, like how objects elicit action representations and also action-related activities. I just wanted to add that so in this literature, they've divided object-related actions into 2 types of actions. So grasp and move actions versus tool use actions. And these 2 types of actions may be differentially connected or involved in visual stream and also concept stream. And also for the grasp and move actions, the orientation and size of the objects would also matter. So like in your stimuli, the cups are all put in different orientations, and depending on people's dominant hand, that might evoke different kind of responses also. So I just thought that maybe when you are tagging your stimuli, you might want to take these into considerations.
Okay.
Probably postdoc work is about object grasping, so you can ask her.
I can give the person's names.
Yeah. Okay. I think to keep time, sorry, Amanda, maybe you can ask offline?
Okay,
Is that
sure.
okay? Yeah, so that we can move on to the next person. Who's next?
So we move on to Ziwing and Marcus.
Amanda just posed a question on the Discord, I guess. That's the best. Yeah.
Okay.
Yep. Once you share the screen and you can go on. Yep.
Okay.
Go
Hi,
ahead.
testing. Can you hear me?
Yes, we can hear you.
Okay.
And we can see the slides as well.
Okay, perfect. Okay, then I'll start. Hi, everyone. I'm Marcus from NTU under Prof. Annabelle, and I'll be presenting today on behalf of myself and Wing. Wing is auditing the course, so I'll be the one mainly presenting. So our title of our experiment is functional connectivity, to investigate the functional connectivity between the amygdala and the prefrontal cortex and see whether this can actually serve as a predictor of aggression. Okay, so why aggression, first of all? So there are my two big reasons. The first is personal interest, because for both Wing and myself, we both studied in, we both worked at prisons in Singapore. So we are quite familiar with a lot of aggressive behaviors like voluntary causing hurt among inmates, or like gang violence related things. And we're quite interested in why this happens. And also more on an interpersonal level, like why do people have aggressive behaviors? Are they more impulsive? Are there impulse control issues? And also, I guess clinically, it can serve to inform like better interventions in future. And also why, another reason is because this project, we need to have some kind of data analysis. So we chose aggression, which is already a psychometric variable that has a lot of neural associations, so it's easier to analyze. Yeah. Okay, next slide. Okay, so some of the background that we found so far is that there's already a lot of aggression research studies online, but they tend to be more with, in the self-report measures or behavioral paradigms, and less interconnected with like more biologically with the explanations. And another thing is that, so while there is aggression behavior with the self-report, there are also some like neuroimaging research done so far on aggression. And it has shown like the amygdala and the prefrontal cortex are quite relevant in this area of aggression. But so far, the reason why we want to do this study is because we realize that they are in silos of each other. So we think that the research gap that we found is that these psychosocial factors are quite separate from the fact on how these neural systems can work. And we want to see, oh, is it possible to predict vulnerability to aggressive behaviors using these different variables? Okay, so far we've conducted some literature review. So it came about 6 psychosocial factors that we think are relevant in predicting aggression. For ease of viewing, all the factors will result in increase of aggression. So increase in stress is increase aggression, decrease in self-efficacy will increase aggression as well. So this is what we found so far. And so, there are 6 psychosocial factors, but for the big 5 personality traits, we broke them down into 2 subscales, which is agreeableness and conscientiousness. Yeah. So then our research question, which is clear, is do these psychosocial factors, the ones that we mentioned previously, predict prefrontal cortex functional connectivity with the amygdala, and is it predictive of aggression and vulnerability? So we have 7 hypotheses. 6 of the psychosocial methods, 6 of the psychosocial factors, and the 2 split apart, the agreeableness and conscientiousness for the big 5 personalities. So these are the hypotheses. I won't go through because I think it's quite self-explanatory in the interest of time. Okay, so how will we go about investigating these findings? So we are intending to use the Human Connectome project, the S1200 version, because there's 1,206 participants, age range from around 22 to 35, and this data was collected around 2012 to 2015. The reason why we decided to use this is because it's open source and there's a lot of data on it, and it covers both like the psychosocial aspects as well as the neuro side. The variables that we are intending to use, and for just time, are as mentioned. These things are like the perceived stress, for example, anger hostility, anger affect, flanker, which is for inhibition. Then the only demographic data that we are able to have, because a lot of it is restricted, so we'll use gender and age range. Yeah, and then also because we want to do functional connectivity, we also look at the resting states of the amygdala and the PFC, the prefrontal cortex. We haven't started data analysis yet, but this is an example of what the data set looks like. It's actually quite a big data set, so it will take time to go through it all, but this is a general guide of how it will work. Okay, so then how are we planning to analyze the data? So on the neuroimaging side, we mentioned that we look at the resting state fMRI so that we can compare the functional connectivity between these 2 regions of interest, which is the prefrontal cortex and amygdala. And what kind of data analysis we'll do is that on R, we're planning to use robust regression find correlations between it, because we can't really use it as we can't say that it's affecting each other, but we can just see what the associations, and also use exploratory factor analysis, because a lot of the variables that we choose might be interrelated, so that might lead to like what's it called, multi-collinearity. So this will try to reduce that fact. Yeah, so that's generally our project so far. Thank you.
Thanks, Marcus. Awesome. I have one quick question. Can you go back to the hypothesis slide?
Sure.
I think, or very quickly, I guess. So everything goes down, the connectivity, all the functional connectivity between PFC and and amygdala goes down, right? Yeah. So then I'm just wondering, do you need to have so many hypotheses? Why don't, because all of them are the same effect? Or, I mean, I'm not saying you need to answer now, but you can think about it. Like when there's only that many degrees of freedom, so to speak, like, you know, the PFC amygdala connectivity, then maybe you can't really have so much, you know, distinctiveness, because everything is correlated. So then how can you be sure that there are 7 different things happening?
Oh, you know, that's why we are planning to do exploratory factor analysis later on. Yeah.
So be careful about that. Like when you make the hypothesis like that, it looks like you don't really have that, you know, much. You can provide a structure then, right, to see how these things relate together. Then it might be clearer. Then the other thing is, if you have only, I've checked, you're taking the resting state data from the PFC and the amygdala from the HCP data, right?
Yes, correct.
So maybe it might be better, useful to also take maybe a control region, a region which you don't expect to be involved, so that you can check. Yeah. So that's one thing to bear in mind. Any other questions?
Yeah. Hi, Marcus. Thanks for the presentation. Very, very concise. Just one practical question, right?
Yes.
Have you tried like analyzing at least one participant, because you're looking at like 1,000?
Yeah.
So it's like it will take a long time probably, but I don't know. Maybe you are just extracting specific functional connectivity. Even if you do that, right? The pre-processing, all that. Have you done it? Tested out?
Well, yeah, unfortunately, we haven't yet done any data analysis, because we actually had issues connecting or logging on to the data set. So I think Wing only got access yesterday. So
so the one thing is first downloading what you need may take time, or even, you know, I would suggest maybe you try it in a small data set first to see how long it takes, and maybe a little bit over ambitious. I don't know, depending on how much compute you have, to try and do all 1,000. But again, I mean, looking at your hypothesis, you may want to be more parsimonious in your hypothesis, so that, you know, don't need that much N actually to look at it.
Yeah, I think that's a very realistic, practical question. To pre-process 1,200 brains, it will take you a month. And you have 2 weeks. Minus weekends, minus whatever. Solution is don't process everything. Just, you know, for the purpose of Brain Hack School, just do a few. And or if you post on Discord, maybe someone has already processed it and can give you sort of, you know, some digested data. Depends on what your goal is and what you want to do. So, but at least, let's say, assuming that, you know, no one has it and you want to start from the scratch so you can get the training. Yeah, maybe bite size, just do a few. Don't need to do 1,000.
Okay, that makes sense.
Okay.
Thank you.
Any others? Okay, if not, then thank you. Let's move on quickly to the next person. Who's next?
The next one I pass to Yu Ting. Yes. Yeah. Can pass my. Yeah. So just share the screen and then you can just go on with presenting. Just.
Okay, so My name is Wu Yuting, and I'm from National Taiwan University. This is the title of my study, Morphological Awareness Effects Across Reading Development Stages. And the following are my keywords in the study. The first one is morphological awareness. It means the ability to reflect on and manipulate the morphological structure of words. And the second one is reading development, because I want to compare the brain activation between across different reading development stages. And the third, apneas. My data the data in my study was collected using apneas. And let's move on to research background. Based on Charles, he thinks reading development was divided into a lot of stages. In addition, most of the theory of reading thinks making a good reading comprehension needs different kinds of linguistic components. And before, most of the researchers think PA plays the most important role in reading. However, nowadays, some people think MA also is also a key component of Chinese reading development. So this is why I focus on MA. And so this is the end of my study. To examine MA-related brain activation across two reading development stages. And this is the group differentiation was based on chose the structure of reading development stages. And I divided the participant into two groups. The first one is grade 1 to grade 3. It is learning to read. And another one is grade 4 to grade 6. It's reading to learn. And let's move on to method. The skills which newly learned from this class as which I use is Git and GitHub and the terminal and the writing scripts in Python. And about data origin, the data was from our lab. The raw data was divided into two parts, typical development and dyslexia. And I just use the data from TD. So this is some information from a participant because I want to compare the brain activation between two different reading development stages. So the two groups has significant difference in age. And I control IQ and the ability of character recognition. And let's move on to Python-based analysis pipeline. I expect in part 1, I will do data check, includes low data, rename stimulus computer condition to make it readable, and exclude incomplete data. And in part 2, I will do preprocessing. Because the FNIR's raw data was collected as light intensity signals, so I need to convert it into OD and then convert OD into HB. And then I will do baseline trimming. And in part 3, it is about statistics. In subject level analysis, I will use GLM to do the analysis and put short separation channels into control variable to exclude non-brain activation signals and say HRF basic function and set the peak time equal to 6 seconds. And then I will put HRF setting into GLM. And in group level analysis, I will use mixed effects model. And in part 4, I will do plotting. The first thing to do is set a channel coded coordinate field and define 3 MA only contrast. And then I will organize the corrected and uncorrected significant channels. Finally, I will show brain activation map visualization. So in the end, The expected output are publicly GitHub repository and a reproducible FNIRS-MA analysis pipeline and statistical outputs. And I will show brand activation mapping for MA specific concept. Testimony is privacy protection and the exclusion of raw data and the subject information. So this is my presentation. Thank you.
Okay, thank you, Yuting. Any questions online? Amanda, go ahead.
Yeah, it's kind of like a naive question. I'm just really curious about the pipeline. Can you go back to the slide where you have the preprocessing steps? Like, I just wanted to ask, like, if you are planning to use the M&E library for your FNIRS data, because I use M&E for a lot, but mainly for EEG, and I'm just curious if the library fully supports all the preprocessing steps that you need.
Because I originally do it on MATLAB, and right now I'm trying to build a new code in Python. So
Wow.
Yeah, I know, but like some of the preprocessing steps in MATLAB might not directly have equivalents in Python or M&E, because at least that's true for EEG lab versus M&E.
Yeah.
So I was just, wondering if you looked it up.
Did you check MNE? Do you know MNE yet? Did you go through the MNE tutorial? I think it's in the Brain Hack School. I think there's one tutorial.
Yeah, but it's mainly for EEG, and I know M&E has some functions for FNIRS, but I have never personally used any of it.
I don't think
yeah.
I don't think Yuting went through that one. I don't know if you want to take a look at it and then see if there's some things that might be helpful. Yeah,
because if that does not work out, you might still need to pre-process your data in MATLAB and then kind of export the data into Python for your downstream analysis, like the GLM or LME. So yeah, you had to look it up.
So my quick advice is you are going to do a very difficult thing. Don't try and do it alone, and don't try to say, just start from scratch. Maybe quickly take a look at MNE or some others and do a quick Google search. Then, someone maybe have already some basic things that you can use, and then start from there. It might be more, productive, easier. Any comments? Kevin has something. I know Darren had a question.
So I noticed that MNE released some kind of FNIRS function several years ago, but because I never used FNIRS, so but you need to go through the tutorial very carefully because for now for myself it looks interesting. Well, this tool can do a lot of things, not just MEG or EG, include FNIRS. So but I have no experience about that. So maybe you can just look at the tutorial and then look at whether other people have their own experience about how that works. Yeah. And regarding your research part, could you describe more about what's is the participant participated in any specific task during your recording of FNIRS? Because I kind of missing that part. You mean you are run a mixed effect model analysis, so that might indicate you have some factors, but I just wonder what's the task you apply to recording the FNIRS data.
it means we will let students to put on a cap, and the computer will show 3 words, and it was the first one. I try to think The first one, they will show a term that's like 鸡蛋, and it will show two terms to choose. It's an 机器 and 鸡, and students need to choose the meaning, which is the same or similar to the items.
Okay, so like meaning recognition from the morphological cues.
Yes.
Is that right? Okay. I think there's more to talk about, but let's take it offline, I think, for time. Darren, you had a question. Is that right? Oh, no? Okay. All right, then in that case, yeah, we can talk offline more, and then we'll move on to the next person for in interest of time. Thank you.
So the next person will be you, Ling.
Oh, okay. 玉玲.
Hello. Can you hear me?
Yes, go ahead. Yeah.
Okay, good afternoon, everyone. I am Weiyou Lin from Graduate Institute of Linguistics and NTU Taiwan. And the title of my dataset is Multimodal Cognitive Workload and the MBAC task for difficulties. Okay. And it is a quite new dataset and from Leon Booth and Matthew Barris and on Open Neuro website. So this dataset integrated EEG, ECG, and pupillometry analysis, but I will only discuss in EEG. Okay, and first of all, I will go over the data overview and theoretical framework. Okay, so this is the MBAC task framework. So MBAC is a standard test of working memory and cognitive workload. So subject must decide if a current stimulus matches the one presented n steps ago. So you can look at the pictures here. If the first letter on the screen is B and the second one is D, and the third one is L, and the fourth one is L again. So that means this L is the same as the previous letter. So the second L is a target. So the participants now have to press the button. And this is the one back set. And if for the two back, the first letter is also B, and the second one is D. The third one is L. The fourth one is D again. So this one, D, is the same as the two previous steps, the same as D. So the second D is the target. So participants need to press the button now. So this is the sequence of one back and two back. And this data set includes one back through four backs. So the stimulus will present 1 second and ISI is 0.7 seconds. So this task needs maintain and updating the information continuously. And also the data set includes the total of 18 participants because originally there are 20 participants, but 2 subjects are excluded because of severe data quality failures and hardware sync issues. And their age ranges from 18 to 48 years old. And there are 6 males and 12 females. And this is the EEG specifications. There are 19 channels and 1 to 20 standard systems. And the sampling rate is 250 Hertz. And the reference linked ear reference, electrodes M1 and M2. And also because it includes EEG, ECG, and also pupillometry, so it has a LSL system to recording and store the information. And this is the protocol summary. So it starts with a tutorial phase with a feedback. So 20 trials at each level, 1 back through 4 back after 60 seconds fixation. And the main experiment is 100 trials at each level and also 1 back through 4 back, but with no feedback. And each level begins with a 6-second instruction screen and with shows remember and steps back. And each trial shows a letter for 1 second and followed by 0.7 blank interval. And so this the file includes event code like MBAC level, key press, match, and response accuracy, and also tutorial flags. And this is a timing sequence. It starts with an instruction and stimulus, reaction, and then black interval, and then the next stimulus. Also, this is the studies regarding MBAC task. But there are some other components in MBAC task like N2, P2, and NSW, but I only focus on P300 here because P300 is directly linked to cognitive sources reallocation, and they also mentioned and discussed in many studies. So, for example, it finds that P300 amplitude diminishes as the cognitive workload increases. So this reduction observed as both frontal and parietal sides. So the effect is often more pronounced at parietal region. And also the second study talks about early and late P300 components. They distinguish between these two subcomponents, early P3 and also late P3, but both components show significant amplitude decreases as working memory load increases from 0 back to 3 back. And also they talk about latency stability. So amplitude is highly sensitive to load, but P300 peak latency is less affected by changes in task difficulty. So, so it shows fewer reliable correlations with workload than amplitude does. So you can see the waveform here, although it's quite small. So the black line is 0 back, and the red line is 1 back, and then the black dot line is 2 back. The 3 dot line is so the black dot line is 2 back. The 3 back is red dot line. So you can see the 0 back line have the greatest amplitude. Okay, and then it's we talk about individual differences in working memory capacity. So because P300 wave also serves as a distinct neural signature that differentiates individuals based on their inherent cognitive abilities. So for example, in the studies of John et al., they have high working memory group exhibit significant larger P300 amplitude across all difficulty levels compared to those with low working memory. And also larger P300 amplitude in high working memory individuals suggest they make more efficient use of neural resources.
Sorry to interrupt. We just have 1 more minute left in the interest of time. Okay, summarize it? Yep, thank you.
Sorry?
You have 1 more minute left. Okay, summarize your pitch.
1 moment left. Okay. Okay, so we just move to my project ends. Because this data set range from 1 back to 4 back. So 4 back is usually considered a very difficult one. So I want to identify the neural breaking point. That's the threshold where working memory load exceeds cognitive capacity, and also compare how this differs between high and low working memory individuals. And then my second aim is to investigate the neural dynamics of the recovery or the giving up phase after making errors. So these are my hypotheses. They may have behavioral collapse and neural attenuation and also interaction effects by low interaction. And also this hypothesis for the M1. And then this is a hypothesis for the M2. Maybe they will, for the high working memory group, maybe they will have a recovery phase. Okay, they rebound in P300 after making errors. But for a low working memory group, they may encounter a giving up phase. So maybe there's a failure of P300 recovery. Okay, and these are my methodology. So this is what I have done. I defined capacity groups and separate them into high working memory group and low working memory group. And for the accuracy rate in high memory group is 64.8%. And also for the low one is 58.6%. Okay. And my EEG preprocessing, in the progress. I'm working on it. So, and first I will do the ICA to do the AD artifact detection. And because this is mobile EEG, so there is no VEOG or HEOG. So I will use FP1 and FP2 as EOG channels to detect the blinks. And also, I will apply the 1 to 30 Hertz filter, aggressive filter for ICA inspect, and then I will use a clean ICA weights applied to the mild filter, like from 0.1 to 100 Hertz bandpass filter to prevent P300 attenuation. And also I will do an epoching for the M1 from 200 milliseconds to 1000 milliseconds, okay, at the stimulus onset. And then for the M2, I would epoch for the trial N+1 epochs characterized by preceding trial and accuracy, like correct or incorrect. And this is my statistical model plan. So it's still, I'm still working it now. So for the M1 behavioral 2x2 mixed ANOVA, and for M1 neural maybe 2x2x2 ROI ANOVA for the P300, and then for M2 recovery or giving up maybe for 2x2x4. Okay, for trial type, post correct and post error, and also for follow ANOVA. Okay, so these are my references. Thank you.
Great. Okay, thank you. Any questions online, people?
I have a quick question. Why the low pass filter is 100?
I'm sorry?
Why the low pass filter is 100 Hertz?
Oh, okay. Because in the previous studies, I found the low pass filter they said 100. So I think maybe it's a common one to set. So and also prevent the P300 attenuation, I guess.
Okay. You probably like to look into a slightly more P300 literature a little bit more. I'm not sure whether that is a good parameter.
Oh.
Yeah,
You mean
just in case. Yeah.
The slice P300.
Yeah. The
suggestion is also to look at the literature or
just look into more literature just in case that the low pass filter is a good parameter for you. Yeah.
Okay. Thank you. Right, for this one. I
Have
guess
you tried it
this
yet?
study.
You said you're using MNE, right? You're using MNE Python.
Yes.
Have you tried it already?
Yes, I'm working on it. I'm trying to be friends with it every day.
Okay. Any questions?
Can I ask? Are you planning to manually reject independent components, or are you using the automatically detecting blink components function in MNE?
Oh, I'll use manually, I guess. So maybe after, maybe after marking, I will look at the result and then I will do it manually.
And the thing about manual rejection is that in MNE, if you're not using interactive plots, it's very hard to view the component activation waveform. Because like in EEG Lab, there's a drop down menu, and you can select component activation, and you can kind of like scroll through the window. And in MNE, you might really run into interactive plot issues. It might not work on your machine depending on your operating system. So if you are doing manual rejection, you might consider exporting the file as an SDT file and do the manual inspection in EEG Lab, because I think the M&E plotting GUI is kind of trash, personal opinion. Yeah. And also, what do you plan to do if no components are rejected? You still might need to do some kind of artifact rejection for that participant, because not all noise is links.
Mm-hmm. Yes. Yes. Okay.
How do like you might want to think about how you might go about that if no independent components are rejected for that participant, and that does not necessarily mean that this participant's data is absolutely clean. Yeah.
Mm-hmm. Mm-hmm. Okay. Okay. I'll think about it.
Okay.
Okay.
Cool.
Thank you for
Thank
your
you.
suggestion. Thank you.
Okay, let's keep the time.
Very much.
Thanks so much.
Thank
And
you.
who's next?
Cindy Caroline. So she's. Do you have a mic? You have a mic. Okay.
Okay, so hi everyone, I'm Xin Ru from Department of Psychology, currently a first-year master student at NTU Taiwan, and my final project topic is EEG signal analysis and classifications of multisensory stimuli using machine learning. Okay, so a little bit background about me. I have no experience in EEG data analysis before, so this project is going to be my first hands-on experience on EEG data analysis, in which the project goal is to gain practical experience in EEG signal preprocessing using Python-based tools. So basically, my project will use a Python programming language, and to explore a machine learning approach for EEG signal classifications. Yes, and not to forget, the goal for the project is to investigate whether EEG patterns corresponding to different sensory stimuli. So the data set that I'm going to use is from Open Neural EEG Semantic Imaginations and Perception dataset. So the dataset consists of EEG signals, of imaginations and perception stimuli from three modalities: visual pictorial, visual ultrographics, and auditory. So basically, the topic of this research is to answer questions that to investigate whether different sensory stimulus modalities, such as image, words, or auditory, produce distinguishable EEG patterns. So whether we can, through the EEG data analysis, we can see that when people look at the pictures, or when people look at the text, or when people listen to an audio, is there a different brain wavelengths in their perceptions? So my data analysis pipeline is going to use MNE Python, scikit-learn, NumPy, and Matplotlib, which basically all in Python programming language, starting from importing the EEG dataset to data preprocessing. So data preprocessing, also including the filtering, the epochs subsetting, and baseline corrections, artifact detections, ERP averaging. Yes, all of these steps is used to improve the quality of EEG dataset before we proceed to the next data analysis. So the next one is feature extractions, which I plan to use a power spectral density to transform the EEG data, to transform the EEG brain data to a number across different frequency folds, so that these features can be read or being learned by the machine learning models. And for the machine learning classifications, I'm going to use SVM and random forest to compare between models. And finally, the evaluations and visualization is to evaluate the model's performance and to visualize the EEG characteristics. So it's going like show three plots across different modalities. So the expected results for this research is to investigate a distinguishable EEG pattern across different stimulus modalities, so that we can know whether different modalities, like when you're looking at pictures, looking at text, or listening to an audio, there's actually a different kind of brain waves in your brain, and to develop machine learning classification models, which is a model that can automatically distinguish or like differentiates between the stimulus that you are looking at or the stimulus that you are getting. Okay. So, and yeah, for me personally, I hope to this project, I can get a better understanding to EEG preprocessing, feature engineering, and machine learning workflows, so that after I kind of like accumulate a lot of more experience on like basic data analysis, I can incorporate more advanced data analysis methods such as deep learning for the EEG decoding, and maybe it can use to be contributed for cognitive workload and attention monitoring research in the future. Yeah, so that's all for my pitch today. Thank you.
Thank you. Thanks, Hiro. Very good. Any questions?
I'm just curious. What features do you plan to extract?
You mean the features?
Yes.
I think maybe it's kind of like looking at the different frequency, like maybe picture was being perceived in some specific frequencies in your brain data, so that different stimulus have different frequency, and these are the features that can be learned by the machine learning.
Okay.
Mm.
And
yeah, go ahead. Yeah.
how many? I might have missed this. How many participants do you have?
Oh yeah, they have 13 participants and 10 trials per participants.
What 10 trials
okay.
meaning?
What kind of? You might want to do cross validation. What kind of cross validation do you plan to do?
Yes, it's a very good question. I've ever thought about this, but I'm still thinking like which kind of cross validations I can do for the machine learning. Maybe if there's any better suggestions for this.
You could leave one out, but EEG has a lot of across participants variability, so that might not generalize well. So you might want to test it out.
Okay. Okay, yeah. I'm gonna find out.
Yeah,
Annabelle had a
very
question.
ambitious project, honestly. I look forward to your results.
Annabelle had a question.
Oh, no, not really.
Oh, no? Okay.
Charlene has a question.
I was just thinking that, so Cindy, you haven't thought about what features to distract to for your subsequent analysis, right? And you mentioned that frequency may be a possibility, but that means that you also need to, I guess your data is in the time domain, so it's like each data point, the microvolt, the fluctuation. So that means that you need to do this frequency transform first, which would be additional steps. So I think maybe you can also think about like extracting the first few, first few 100 millisecond data point in the first few 100 milliseconds, because we kind of already know that visual and auditory stimuli have different kind of evoke potentials, and that differentiate the most in the first, let's say, maybe 200 milliseconds. So like if this is like a practice, maybe you can start with that also.
Okay. Yeah. Okay, thank you for the advice.
Yeah, it's a very complex. Obviously, there are so many features and a lot of
Or
things.
try both time domain and frequency domain features and
If
put
she
it in
had
a model.
time.
and see which one improves the accuracy or something like that.
Yeah. Last thing, I just wanted to clarify. You said there's 16 subjects. 13 subjects, and each one has 10 trials.
Yes, 10 trials.
10
That's
trials
actually a small
of
sample
what?
size.
10 trials of each stimuli?
Yes, it's stimuli.
Okay.
3 stimuli.
So that means it's like 30.
Yeah.
Okay. All right. So that should be okay, right?
130 might be slightly small. Your model is like SVM or logistic regression.
SVM.
Yes.
Yeah.
Maybe a bit small. I'm not sure. You have to test it out.
But the
Yeah.
I think can't hear what Joshua is saying.
Oh, sorry. Yeah. Ting can't hear what you're saying. Well, Ting is suggesting, sort of like what Charlene was saying, that you can use the first few 100 millisecond because we know pretty clearly, about the visual and audio signals already. So you can use that epoch or that time window itself as a way to validate, like, the frequencies for other windows.
Yeah, but you have to think
Which-
about how to
Yeah.
represent the time domain, time series as a vector for the SVM.
Use
So
the
that's
mic.
feature engineering.
There's a
Okay.
mic there. Yeah. I
So-
think there's
so,
last
yeah.
question, last comment, and then we have to move on. And you can take it offline.
I just
last comment.
have a very quick browse on the data sets. So it's more than 10 trials. So yeah. So it says like 5 repetition for each stimulus and in each block, and there are 10 blocks. So
10 blocks with 5 repetitions. So it's a lot more.
Yeah. So
that's sufficient.
yeah.
Okay.
So yeah, sounds good. Yeah. So but to be careful about the filtering, because you're going to use the frequency domain measurement, and filter might wash out something you want to observe. Yeah.
Yeah.
Okay, thank you.
But the first few hundred milliseconds for the later time points, that's really very good, because we have some sort of ground truth, or we already know some things about the way stimuli are processed. So you can use that as leverage to validate, instead of just purely from data mining kind of approaches. Yeah.
Okay.
Okay.
Thank you,
Awesome.
Professor.
Very awesome. Thank you. Next, who's next?
On to Ling, and she has the mic as well.
Could you?
Okay, good. Then
Okay,
go ahead.
So hello everyone, my name is Jill, and I'm from the same lab as Wu Yuting. So we have kind of like a similar data and similar like processing. And so I want to see the brain behavior analysis among children with and without dyslexia. So the method we use is to use the FNIRS, functional near infrared spectroscopy, and to examine the neural correlates of children with and without dyslexia. Specifically, we want to identify the brain region differences during the morphological awareness task between these two groups. And for the behavioral measure, we want to examine whether the neural activation is associated with their literary performance. So specifically Chinese character recognition and morphological construction. So this is what FNIRS looks like. And this is like 2 independent tasks. So totally we have 3 tasks, and they are, the first one is FNIRS, which is machine-based, and the second 2 ones are using pencil and paper. So here I have 60 children, half with and half without dyslexia. And before I start this project, I have pre-extracted the HBO beta values during the FNIRS task among totally 32 channels. And behaviorally, I have also, we have got the morphological construction task and character recognition scores. So initially, based on a group level analysis comparing these 2 groups, 3 target channels were selected because they show group differences. The first one is channel 6, which survived correction at FDR correction, but if it's not corrected, there is also channel 16 and channel 21. So based on these findings, my initial plan was to focus on these 3 channels and examine their relationships with behavioral performance using Pearson correlation. So here is where the class, where I try to apply what I learned from this class. The first one is to visualize the data and write the script in Python to calculate. To correlate to calculate the Pearson correlation and do a whole brain exploratory analysis. So first, this one is just kind of like for a better view of the data. So this one, when I move, I can see the score of each participant. And then I write a code to calculate the brain behavior correlation, but nothing is significant. So after a discussion with the TA, she pointed out some probable like suggestions. The first one is selection bias. So these 3 channels were selected because they are already significant. They are already different among the groups. So this may like increase the significance. And I may also miss other other channels. But I also asked and discussed it with with the members of my lab, and they say like it's kind of okay because morphological task and the FNIRS task, they are independent and they are not related. So perhaps I can accept this non-significant result and try other behavioral measures to see the correlation. But there's also another question, which is a logical mismatch, because the LME group condition effect does not necessarily entail a brain behavioral association at the independent level. So this is why I want to do a whole brain analysis to use Python to analyze all 32 channels to avoid this pre-selection bias. So I make a 8x4 grid, and still no channel survives the FDR correction. So it's kind of look like this. There is no clear linear brain behavioral relationship, and in total it look like this. So the next step I may do, I still learn something from this, although it is not significant. So first one is I know how to write in Python, and I understand the possible bias of double dipping. And so in the future, I may explore other non-linear models, such as log transformation, and if possibly, I would increase the sample size or try correlation with other behavioral measures. So this is my presentation. Thank you.
Hey, thank you. Questions?
It sounds like you've done the project already.
I've done this is like for now I've done until here, but I'm thinking about how to explain this because nothing is significant. Yeah, so I'm
That's quite normal. I'm wondering if you are working with the other student.
We are using the same data, but this is kind of like both are part of our thesis. So we are.
So it's better for you to work differently rather than to work together.
Yeah.
Okay,
Well,
that's fine. Just checking. Yeah, just making sure that that's okay.
Thank you.
Okay. Any other technical questions or suggestions?
I have a quick question. Because I probably missed that. You show a lot of correlation across different channels. But from my understanding, FNIRS is a time series signal. Then like which part of the signal you are correlated with their behavioral
You mean
performance?
which channels or?
Which timing, I guess?
I've done the pre-processing in MATLAB, so I think it is like a 5-second period, and
Is this the VO signal, the HBO2 signal?
yes.
Over a 5-second window.
Yes.
Oh, so that's what it is.
Okay, thank you.
Thank you.
Yeah. Is that standard?
Yes, I think yes, it is.
Okay. I don't do FNIRS, so unless they're FNIRS people, or you can look up the literature or post to make sure that I believe sure it's standard, but just to make sure.
Okay, thank you.
Okay. All right, I think that's it for this question. Thanks, and let's move on to the next one. Exciting works.
Pass it on to Isaac Huang.
Can you see my slide now? Okay. So Good afternoon, everyone. My name is Isaac Huang, and I'm a first-year grad student from the Department of Biomedical Engineering. So a little bit about my personal research. It relies heavily on biological wet lab experiments. So I also have close to zero experience on these related topics, and hopefully I get to learn a lot from this project. So today I'm introducing an open source Python pipeline designed to explore probably one of the most fundamental trade-offs in modern neuroimaging. So as researchers, we are constantly forced to choose between the spatial resolution of fMRI, which tells us where things happen, and the temporal resolution of MEG, which tells us when things happen. Now, this pipeline is explicitly built to bypass that trade-off. And wait, does this block anything? Okay, sorry. So I will try to build a unified computational workflow that ingests separate multi-sensor open data sets and probably decode complex human action, and uses transfer learning and pattern similarity to map brain dynamics in space and time simultaneously. So to pull this off without the friction of under-standardized data format, we leverage two powerful complementary open access resources that would be the human action dataset for fMRI and its sister dataset, the HAD-MEG. So these datasets contain brain responses to numerous video clips from 30 participants, and the video clips encompass 180 human actions categorized and offers a comprehensive coverage of complex activities in daily life. Now, what makes this combination incredibly unique is that both cohorts were recorded using the exact same experimental paradigm, containing nested hierarchical action labels, which means a subject watching a video clip of, let's say, someone surfing should be coded identically whether they are sitting inside an fMRI scanner or wearing an MEG helmet. So furthermore, because both datasets follow the bit formatting, we can supposedly, I would try to build an automated programmatic data ingestion loop that treat these distinct physical measurements as parallel features of the exact same cognitive process. Now, before we can execute any machine learning, we have to align these raw multi-sensor signals into a shared computational space. This slide maps out the core pre-processing pipeline I'll be writing in Python. So for the MEG data, we will utilize ME Python to filter the signals, remove biological noise, like probably caused by heartbeats or eye blinks using automated components analysis, and slice the continuous data into standardized time windows around each action. So in parallel, for the fMRI, we will focus on probably Nylern or Nibabble to extract static 3D activation maps. This should give us a localized snapshot of which brain networks are engaged for each specific action condition, preparing both data sets for the next stage of analysis. Now, with the clean data aligned, the pipeline splits into three major engineering objectives. So the first two pillars focus on the signal classification and the algorithmic portability. And first, in the decoding engine, we will extract standard frequency bands like the alpha, beta, or gamma from the MEG signals. We will then feed these features into a machine learning classifier, like perhaps for now I'm considering like SVM, training it to identify exactly which action a subject is perceiving based purely on their brainwaves. And the second pillar addresses a massive bottleneck in neuroengineering. It would be the hardware accessibility. So as we might know, MEG systems are more rare and expensive, while EEG is more portable, available, affordable, and accessible. So we will use a technique called unsupervised domain adaptation to align the underlying pattern of both data sets, trying to allow us to test whether a model trained exclusively on high accurate magnetic signals can perform zero-shot decoding on a noisier real-world electric scope signal. Now, after that, the third pillar would be a more, I would say it's the core scientific fusion engine of the whole project. Like, how do we mathematically link the 3D spatial voxel volumes with a 2D time verifying sensor. So we would use this RSA, representational similarity analysis. So instead of trying to map physical sensors directly to the brain tissue, we convert both data sets into abstract similarity spaces. We calculate how different the fMRI spatial patterns are between different actions, and then we calculate how different the MEG temporal patterns are across a sliding window of time. By correlating these two similarity maps, we essentially translate both datasets into a shared language, and this output is a highly precise timeline showing us exactly how many milliseconds after a stimulus occurs that the brain localized a spatial network fetch life. It looks like I'm taking a bit too much time, so I might as well skip this page. There would be, these are just some simple examples, and there would be a lot we can explore once we get to the bottom of this. But for the main conclusion, the ultimate goal is not just to generate a set of research figures, but to rather deliver a robust and reusable tool. So by the end of this project, maybe, or this might as well be a rather long-term project, I hope I would be able to maybe deliver a fully documented open source GitHub repository, and it will then include a interactive Jupyter Notebook tutorial and probably a completely containerized Docker. And
last minute.
Or 1 minute, or should
Oh,
I?
you can finish up.
But yeah, that's pretty much it. So thank you for your time.
Okay, thank you. I have one question. Is so regarding the data that you are going to work on, I want to clarify that the subject was simultaneously.
No, it wasn't.
It's the same subject.
Yeah, the same person doing the same thing, but
in different sessions.
fMRI and the EEG weren't simultaneous.
I see. So I think that, well, that could or could not be a limitation that you want to think about for what you want to do, right? Obviously, even if I'm looking at the same video second time, it's not the same.
Yeah, I know. Yeah. There was actually this other data set I came up with. It was like recording sleeping states, both simultaneously, fMRI and MEG.
Yeah,
might
but it's
also look into that. But yeah, that's a problem. The problem with the sleeping data is that the applications, it's more like.
I agree. Yeah.
So
I think the saving point here is that so we look forward to the tools, but at the end of the day, I think the more compelling thing is the RSA at the end, because instead of trying to say that these are simultaneous signals that you can link, you derive some sort of a more abstract, similarity space kind of states that you can transform or between the different modalities. That in itself is already, but you're not saying anything about simultaneity. You're just saying that if I see this brain state that is I know has something to do
probably
with
predict the action.
And then how therefore this, if I have this action, what will I predict for the MEG state? And therefore how does a brain state predict the MEG state, so on and so forth, given the center metric, which is the RDM, the dissimilarity matrix, right? Something like that would be, I think, quite valuable. Yeah. So think about, but very ambitious. You probably won't be able to do it in 2 weeks. I don't know.
Yeah.
So we'll see
probably
how
just see how far it goes.
Yeah. How other questions, comments? Okay, if not, let's quickly move on to the next one. Thank you so much.
I'll pass it on to Xingzi. Yep.
Wait. Oh. Okay. Sorry.
可以哈，可以吗？
Okay.
好啦，可以。
Okay, so I'm Xingzi Lin, and I'm a first year master student in NTU TW Psychology. My project pitch here today is kind of a variant of my master thesis, and I want to explore the temporal awareness in the context of multisensory integration and the neural correlates of of altered temperature awareness in autism. But the background is like the speed of light and the speed of sound is different. And also the processing of our visual and auditory systems are, the processing speed are different. So I'm interested in how we perceive cross-modal sensory experience with different time scales as one event. And it's the topic I'm gonna explore through a multi-time scale approach. So I'm gonna bring some background of my project. And the first is that our response to multisensory stimuli are not just simple summation of responses to any sensory stimuli. For example, we have a super additive responses, which means the effect of multisensory integration is larger than combined unimodal responses. And the other one is the additive responses, meaning that the effect of multisensory integration is smaller than the combined unimodal effects. The figure shown here is from an AG study, which tells us that the power changes of multisensory integration of audio-visual stimuli versus sound of unimodal conditions. Yeah. And about this, it's from mid-case layer studies. They found a nonlinear effect of multisensory integration. Here I abbreviate multisensory integration as MSI, and which is the oscillatory power changes in alpha band. And it suggested that alpha suppression index heightened integration load, kind of like additional mental resources when perceiving cross-modal audiovisual stimuli simultaneously. In this study, the task is in the context of speech, but for my own project, the task would be quite a simple one. So I think I can explore what will be different from the result of this study. And next, it has been suggested that different time scales reflected in different oscillatory frequency bands serve to route different information types through cortical networks. And it also has been found that individuals with autism spectrum condition has older multisensory integration time window. However, very few studies investigating multisensory integration on this population through this approach. So my ends are to compare the MSI effects between these two groups, and I mainly use time frequency analysis to explore the MSI effects between different frequency bands. Yeah. And so for my methods, the dataset I'll use was downloaded from OpenNeural, and it consisted of 3 groups: TD, AAC, and AAC, then their siblings. And I'll decide whether to include siblings in my analysis. So it is an AG dataset containing some useful phenotypic information like the above. Okay. And the task the participants did was the audiovisual simple reaction time task. The stimulus type consists of visual stimulus alone or auditory stimulus alone, and combined audiovisual stimuli simultaneously. What participants should do was just press the button as quickly as possible when they detected any stimuli. I haven't found the providers of this dataset publish their study using this dataset. So I think this allows me to form my own hypothesis, like I just mentioned earlier for this project. So for my analysis, I'm gonna use MATLAB mainly and for preprocessing, I'm gonna use EEG-Lab and ERP-Lab. And for time frequency analysis, I'm gonna use field trip. And specific process, I would refer to these 2 paper. Okay, and so I'll after analyzing this data, and I'll visualize the result like the right side figure in the figure, and compare my result with the result in this paper. Yeah, and this is what I've done so far. So that's it for my project.
Okay, thank you so much. I'm going to check you said that they haven't published yet.
They
the data?
haven't published their study using this data set.
Oh, but the data are open.
Yes.
Okay, just make sure it's okay, right?
Okay, it's okay.
So generous. Okay, any questions or recommendations? No? Yeah, Kevin?
Yeah, just out of my curiosity, what would you going to observe based on your hypothesis that autistic participants have like a different integration time in the audiovisual condition? What based on that, what would you?
Not sure what happened. I can't
You-
hear all of a sudden.
Yeah, what would you expect to observe that in your results?
Observe. First,
In what kind of neural marker would you let you infer as the indicator of like longer or inconsistent cross-modal integration? Yeah.
first I'll just visualize the figure, and because I'm not sure I'll do behavioral analysis, so I'll first just visualize the power spectrum. Yeah.
Yeah, okay. For this given time frame, maybe that's quite reasonable. Yeah. Okay, thank you so much. And let's move on to the next
Next one,
president.
we'll move to Singapore for Isaac Teo.
Oh, did Annabelle have problems? Annabelle, you have problems? Can you hear us?
Can you
or-
hear me?
Yes,
I
we
can't
can hear you. We can you hear us? You can't hear us. Oh no, Annabelle cannot hear us.
Is my mic working? Yes.
Yes, so others can hear us, but Annabelle cannot. So maybe Annabelle has to
I don't
set-
know what happened, but turn on my captions so I can see what you're saying.
Is this mic working? Can you guys hear us? Jane?
My audio is fine.
Mine is working also.
Yeah, we'll have Annabelle has to sort out, but I think we just carry on in the interest of time. Oh, reloading. Okay, so Grant had the same issue, but if you log out and log in again, it helps. Isaac, if you can, please go ahead. Yeah.
Yes, Prof. Is my screen shared? Sorry, just to check.
Yes, we can hear you, and the screen's slides are there. Yep.
Thank you.
Yes, yeah.
Good afternoon, everyone. Good afternoon, Prof. and fellow classmates. I'm Isaac from NTU SG. And today I'll be presenting about my project on cluster analysis and machine learning prediction of brain age and its associated phenotypes in autism. So I'll go through a brief overview of the current literature, and then I'll talk about the methods of my study. So very briefly, autism is a neurodevelopmental disorder that is characterized by social communication impairment, as well as restricted interest or repetitive behaviors. And one line of research uses cluster analysis to identify different profiles of ASD. So for instance, we can distinguish between high and low functioning individuals. We can group according to symptom severity or from differences in neural profiles, among many other domains as well, from language and behavior profiles. In terms of the neuroimaging side, structural MRI and functional MRI are rather extensively used to investigate autism. And one line of research is in calculating what we call brain age gap. So essentially, it is using MRI data to generate a prediction of one's brain age. With this predicted brain age, we then minus away the actual chronological age of this individual, and from there we get this brain age gap estimation. So this brain age gap is useful to assess neurological conditions such as Alzheimer's disease and ASD. Specifically, it assesses advanced versus delayed brain development. So for instance, if we have a teenager who is 15 years old and he's diagnosed with autism, using his MRI data, we can generate a prediction of his brain age And if the predicted brain age is 20 years old, then the brain age gap will be 20 minus 15, so 5 years, which means to say that this individual has an advanced brain development of 5 years as compared to his cognitively normal peers. Alternatively, if the predicted brain age is 10 years old, then his brain age gap will be negative 5 years, so this individual would have delayed development. So in terms of brain age gap with ASD, we see that there is a delayed brain development in ASD patients as compared to healthy controls. This is on a group level basis comparison. But within the group itself, within ASD patients itself, there are two subgroups. So one group have advanced brain development, and then another group, some individuals have delayed brain development. And when studies try to associate the phenotypes with these subgroups, we see that there's some discrepancies in findings, whereby one study found that advanced brain development is associated with greater ASD severity, whereas another study found that most advanced brain development show lower severity instead. So why this might be the case is probably different data sets, different neuroimaging modalities used, as well as different ways in which they classify brain age gap. So the first study, they Z-scored it, and anybody with a positive brain age is deemed to have advanced development, anybody with a negative brain age deemed to have delayed development, whereas for the second study, there's another layer of balanced development there. So these differences might have caused these differences in results that we see across associations with phenotypes. So although brain age gap might be useful to stratify ASD profile, no study as to date, to the best of my understanding, has performed cluster analysis with brain age and associated phenotypes. So my study, I'm looking at using the ABIDE dataset, and we'll be using the data there to predict the brain age of each individual. From there, with the brain age gap as well as the phenotypes of each individual, I'll perform a cluster analysis, and we should be seeing that there's more than one stable cluster forming, which would indicate that there's distinct brain age cognitive symptom profiles among ASD individuals. Subsequently, I'll then use the structural MRI and resting state fMRI data to predict cluster membership, and we should be able to do this above chance, which would indicate that the identified cluster subtypes correspond to distinct brain profiles. So exactly how I'll be doing it, first step, from the ABIDE data set, I'll be taking the cognitively normal controls data, using their MRI data as well as their age. I'll train a support vector regression model, and then we'll test the model. From there, we should be getting a low MAE, low RMSE, as well as high correlation between the predicted and chronological age, which would mean that the model that we have trained is rather accurate in predicting one's age based on the MRI data. So this model is good to go, and then now we can deploy it in the ASD population. So from the ASD data, I'll get their structural MRI and resting state fMRI data, I'll pass it through the trained model, and will derive the predicted age. Subsequently, I'll calculate the brain age gap by using the predicted age minus away their chronological age. So with this brain age gap, as well as their ASD symptom scores from the ADOS test set, and their cognition scores in terms of verbal IQ, performance IQ, and the demographic variables, age and sex, I'll perform a cluster analysis using Gaussian mixture model, clustering model, and from there we should be getting clusters of different profiles of ASD. I haven't run the analysis yet, but ideally it could and should look something like this, whereby maybe one cluster is a cluster of females with delayed brain age, with poor social communication skills and low verbal IQ, and maybe another cluster is a cluster of males with advanced brain age, high performance IQ, and with more stereotypical behaviors. And hopefully I'll be getting a few cluster groups with different characteristics. And finally, using the MRI data as well as their subgroup membership clusters, I'll train another SVM model, and then we'll test the model, get the R-square and MAE. And from this model itself, it should demonstrate that simply by using your MRI data, we are able to predict which subgroup do you belong to. So as a whole, the study, we would be able to identify that there are distinct brain age cognitive symptom profiles, that using MRI data itself, we can predict a profile membership above chance. And so together, brain age gap as well as MRI can serve as potential markers for the stratification of brain behavior profiles in ASD. That is all for my presentation as of now. Thank you.
Okay, thank you. Thank you, Isaac. Awesome. Questions from audience? I have one, which is I was wondering, you're trying to use the brain age and the ADOS scores, right? Is that right? Together, and then like as a combined variable set, you want to see if there's clusters of different ASD people use when doing it this way, when combining the scores or using all the scores like that. Is that right?
Yes, that's right.
Okay, so then I'm wondering if it is a valuable thing to actually just see if just with the ADOS scores, you already can cluster people, cluster the ASD, and then ask how much more does the brain age distinguish or revalidate, you know, the ADOS score classification. You know what I mean? As opposed to combining everything and then forming clusters, because what that ends up is like you will find clusters that have the combined scores, right? So, but then does that, you know, I'm not sure. I think that's useful, but then I think also from a mechanism, from a clinical or application point of view, I think a lot of clinicians would like, I'm using my ADOS score anyway, right? So maybe I don't need to scan the brain, but maybe your data will be able to tell clinicians, hey, no, ADOS score is not enough. You know, maybe you need to scan the brain, because then when you have the ADOS score, then plus you have the brain, you can, you know, diagnose the patient a little bit more precisely. I think there will be a promising contribution on that front from this work. Yeah.
Understood. Thank you, Prof.
Yeah.
Yeah, adding on from what Josh mentioned, I guess that's the question now, what's the clinical add-on with your brain age data? And one of the things that may be interesting is probably not possibly answered by whole brain analysis. That's what brain age is doing, right? Is there a way to actually cluster out networks? Because one of the things, if you just look at the ADOS, right, can it classify high functioning to low functioning, or individuals with special abilities or something like that versus low functioning? Because ideally you would be seeing some differences in the and in development wise, like because brain age is like an overall brain development, whether there's delay or there's advance, that's what you're trying to see in the population. So what's the add-on utility? Maybe something to think about. But in terms of this exercise, a practical thing is like, have you run through something to look at ready?
Not yet.
Okay, how many subjects in the ABIDE data are you looking at?
For the brain age prediction training itself, I think I'll use all of the data points, which is about 1,000. And for the autism, the clustering wise, is about 236 participants.
Yeah, that's still a lot, right? So have you done this before? Like, have you done image analysis?
I did it in R, but not this project, but with a different, using the same modality, like brain age wise. I've done it before in R. So yes, I think it is possible this time.
Are you doing it post-process data? So you're not analyzing from
post-process data.
Oh, okay, that's okay. So yeah, already post-process, and then you're just doing the SVM thing to build your model, and then you then do the 200 something ABIDE data. Is that also from scratch, or is that?
It's all pre-processed data.
It's all pre-processed.
Yes.
Oh, so then it's all more values that you're looking at, right? Are you just running it in R, or are you developing Python packages to?
I'm just running it in Python.
Okay. But if what is the brain imaging part that you're doing actually at this point?
Using structured MRI and resting state fMRI data to run machine learning algorithms.
Yes. So are you pre-processing them? Are you learning any of those?
Oh, not in this project. Yes.
Anyways, I'm just trying to understand like how much time you would actually need. So if it's already pre-processed, I mean, you missed the part of learning the analytics of that, but maybe you can pick that up later. But, just to see if it's feasible, probably it's more feasible because it's all process data and just modeling, right?
Yeah, I think one way I could add on to that learning portion would be in terms of the after I cluster, and maybe if I'm able to identify differences in networks between different clusters, I think that would be an added value, as what you mentioned just now.
Yeah, but I'm not sure how you could do that if it's all process data. You know what I mean? You actually get just one value to represent your brain age. So it's not really looking at different networks within the brain. But this is something food for thought. You know, what's feasible and what's not, because it probably is not feasible for you to do everything from scratch. But you may want to think about in the future. Yeah.
I have a suggestion is perhaps, Isaac, it might be educational to try one brain age person from scratch, from the brain data, and then see if you can get the same brain age number that those derived scores already give you. So sort of a replication just for yourself. If you can even just do that, then you know, yeah, the process that's involved in all the whole analysis, which you can pursue later on. So this might be a reasonable thing to do, you know? Yeah, and you can factor that into the project as well. So like you try one or two, and then plus the fact that you are running these clusterings as well in parallel, sort of. So you get the benefit of both the learning the imaging part and also the sort of the overarching bigger project. Yeah, that's just one suggestion. You can think about it, see how you want to navigate that. Yeah.
Thank you.
Okay, other things? If not, let's move on because we still have a few more, but we're almost there. We're moving forward. Let's next person.
We're passing on to Xing Qi.
Xing Qi.
Hi, I'm Xing Qi. Wait for a minute. Can you see my screen? Okay. I'm a first year master student in psychology. I'm a beginner at handling brain data, and my research interest is investigating social cognition in children with autism spectrum disorder using fMRI. I make some goals of this final project. I want to make use of open source data to get experience of doing fMRI data analysis. And at the same time, I want to understand the concept and purpose of each step. So for my final project, I want to do a replication study with a focus on brain activation. Taking my research interest into consideration, I want to answer a question about which region of the brain has significant activation during social cognition tasks. I found the data from OpenNeural. I chose this data because there is fMRI data during social cognition task. It is a raw data, so I can use it to practice pre-processing. There are only 10 subjects, so I think the burden will not be too heavy. All the subjects are adults with typical development because I couldn't find any data set that is autism spectrum disorder. According to the original paper, the author said it is a multi-band multi-echo fMRI. It is said that it can optimize the temporal signal-to-noise ratio. To handle this kind of data, the author used multiple packages and softwares, and their results show that these areas have significant activation during social cognition tasks. So I think that kind of multi-band, multi-echo data may require custom pipeline, but I still want to use MATLAB's SPM because that is what I've learned. And also, I found that the repetition time is 2 seconds, which is the same as a traditional one. And there are 5 echoes, and I would choose the second echo because the echo time is the closest to my professor's data. I'll skip the details of my pipeline because of the time frame. And the expected outcomes is a figure like this down below. on the left side to show what are the regions that have significant activation during social cognition tasks, and are they the same as the authors? But I think different results are highly possible because of the multi-band, multi-echo issue. And this is what I've done so far. One of the subjects pre-processing. So actually, I have 3 questions I want to discuss with you. The first is, I know that script-based tools like Nidern is more friendly for open science, but actually, I'm a beginner at both in fMRI data analysis and programming, so I think I will prioritize SPM GUI. And the second is, just now I just decided to do the whole brain analysis because that's what I've learned. But actually, the author get their results from ROI's analysis. And the last one is, I think, the most important. What should be my pre-processing pipeline? Let's see the next slide. There are 3 kinds of pipeline. The left one is the easiest one I've ever seen. The middle one is another kind, and the left one is from the original paper. Actually, I don't plan to do the author's pipeline because I haven't learned those specific packages yet. So my next step will be to get a deeper understanding of these pre-processing, the author's pipeline, and so as to decide which pipeline I will use, the left one or the middle one. So if any of you have experience of the author's pipeline, your feedback will be very much appreciated. And that's it for my pitch. Thank you.
Okay, thank you, Xinqi. This is classic fMRI pre-processing issues. So we will start a channel, okay? And then you can ask all your questions and then we can answer there.
Okay, thank you very much.
But yeah, Annabelle had a question.
No, just a comment. I think this is great because this will be looking at reproducibility, right? And also, if you introduce a different thing, if you don't want to do the author's pipeline, it'll be interesting, right, to compare the different software on the same data set. But just to make sure you have all the data set, you have T1, T2, and all that. I mean, you have.
Do the data have T2, T1, EPI, all this?
Yes, yes, yes.
They do?
Yeah,
okay.
they do. They have.
I mean, they have multi-echo.
Yes.
So I would assume they have quite a lot.
Yeah, so you just want to make sure whatever is needed. I don't know, like especially the denoising part, right? Field maps and all that. If they provided it in their data set, then you see what is feasible. But it seems like they did quite a lot of cleaning, right? and all that. So don't know what your pipeline, maybe to see how robust the effects are, right? Maybe you have to do all these massaging to get the finding, but it'll be interesting. Yeah. Yeah, do sign on to that channel.
Do we have a fMRI pod, pre-processing pod fMRI? Yeah, make noise in there, you know.
I have. Nobody replied.
Nobody replied. Your noise is not noisy enough.
I'm very sad.
Make more. Oh, really? Okay, people. Okay, William is going to reply to you. He's going to solve all your problems, okay? Because he's also doing the same. All right. Yeah, make more noise. I think people are maybe just haven't warmed up yet. Don't give up, okay? It will be there. Okay, very interesting, but we'll take it offline and we'll solve that. Next,
pass it on.
Thank you.
Chelsea.
Chelsea.
Hello.
Yep. Hi. Okay.
Hi. Am I audible?
Yes,
you are.
Okay. Great.
And go ahead.
Okay. Did my screen show? Yeah. Okay. So. All right. Good afternoon. I am Xuan. You can call me Chelsea, and I'm a first-year graduate student here at the GIPMS almost mispronounced myself. Okay, my project pitch is titled A Configurable QC Workflow for Trial-Wise Task fMRI Metadata. So the main idea is that before we move into deeper neuroimaging analysis, we need to make sure the metadata layer is inspectable and trustworthy. By metadata, I mean the trial level information that connects events, behavioral responses, stimulus features, and eventually fMRI analysis itself. So I want to build a lightweight workflow that checks whether trial-wise metadata are complete, consistent, and ready for analysis. So, okay, managing the time. So, this motivation comes from working with an auditory/music fMRI dataset. I'm working with that dataset for my master's thesis. In this kind of task, a participant may first see a fixation, then listen to a probe melody, make a yes or no decision, and then listen to an outcome melody. So every trial creates several layers of information: the timing of the event, the stimulus features, the participant's decision, the reaction time, the outcome information, etc. And the corresponding run, of course. And the important point is that task fMRI dataset is not only brain images. The brain images only become interpretable when they are correctly linked to trial level metadata. And this project was inspired by the practical difficulty of making those links visible, complete, and consistent. If you know, you know. So when working with real task fMRI data, the structure can become complicated very quickly. So there may be original scanner files, behavioral logs, stimulus information, raw data folders, pre-processing outputs and reports, or quality control derivatives. Even in this simplified data tree, we can already. Sorry, did anyone say anything? No. Okay. Even in this simplified data tree, we can already see that the data are spread across multiple layers. So imaging files are only one layer of the story. To support later analysis, we also need to understand how subjects, runs, event timing, stimulus variables, decisions, and reaction times line up. So the problem I want to focus on is that metadata errors can be quiet but costly. So for example, a table may have missing required columns, duplicated subject run trial rows, unexpected conditional labels, missing or impossible reaction times, mismatched stimulus and response rows, or even trial counts across runs. So some of these problems may not immediately crash a pipeline, but they can still distort downstream analysis, reduce reproducibility, and make the data set harder for other people to understand or to reuse. So my clear goal is to build a lightweight, configurable Python workflow that can inspect, validate, summarize, and report trial-wise test fMRI metadata before deeper analysis. The core design is to make the workflow configurable rather than data specific. So the input would be, say, an event, behavior, or stimulus metadata table. And then the user provides a small config file that defines the key columns. For example, the config files tells the workflow which columns is the subject ID, which column is the run number, which column is the trial number, which columns are stimulus features, and which columns are responses or reaction times. And then the quality workflow validates, summarizes, and visualizes the metadata. And finally, it outputs a report with summary tables, figures, and warnings. I don't want the script to hard code only one experiment. Instead, the user tells the workflow which columns matter, and the same quality control logic can be adapted to different task fMRI metadata tables. So, my time. So for public demo, I don't think I am going to include private lab data. Instead, I will generate small synthetic metadata tables that mimic the structure of trial-wise task metadata. So the code excerpt here shows the basic data, unreal subjects, fake runs, and trials. And for each trial, I generate a stimulus feature, a decision such as yes, no, or missed, and then an outcome feature and reaction time. And so the generated files will also include something like events demo TSV and participants demo TSV. So the synthetic data are structurally realistic. Yeah, so but they're not used to make scientific claims. They only demonstrate that the workflow can load metadata, run checks, and produce a report while keeping real lab data private. So yeah, and after generating or preparing the demo metadata, the user can run the quality control workflow from the command line. So the script, for example, takes 3 main inputs: the event table, the config table, and the output path for the report. And expected QC checks include whether required columns are present, whether there are duplicated trials, whether decision labels are valid, whether missing reaction times only happen when expected, and whether the number of trials per run matches the expectations. Output report would include summary tables, QC figures, and human readable warnings. Yeah, so it should make the potential problems easy to see before deeper FMR analysis. So from demo workflow to real world validation, after this stage, I see a possible extension, which is to, so the current stage is to build the workflow using synthetic demo metadata and make sure the pipeline works. And then as a next step, I privately test the workflow on the motivating lab data set that I'm working with my master's thesis, without including the private data in the public repository. And after that, I would like to apply the same workflow to a public bids task FMI data set, which would test whether the workflow can adapt to real open data using a different config file. In the longer term, the QC outputs could support later replication or modeling work by making sure the fMRI data are analysis ready. And yeah, so the remaining parts of this slide are how my project maps directly onto the BHS modules. I'll skip that for now. Yeah, and backup slides. If someone is curious about the full synthetic data generator, this is part of the code, part 1, and then part 2. And that is all for my pitch presentation. Thank you so much.
Okay, thank you. Okay, Chelsea, this is going to be important work for a lot of people because we all know that open data is a mess. And I mean, frankly. So not on purpose, but it's just the way it is. So yeah, we look forward to, whether you can help people out with a tool that can solve that. Yeah.
Okay.
But it looks like you've got it done. Do you have that sort of script already, or?
I did. I checked with Jennifer's data. Jennifer, the 学姐, the 学姐 who created the data set I'm working for. So but I haven't yet chosen which open data set to to use. Right? Yeah.
Okay, so you have the your pipeline. You've applied it on your own thing, but you haven't ventured to the open data set yet. But obviously, that will be the next step. Is that right?
Yes.
Okay. Right. Any questions or recommendations?
Yes, this is great. I think it's really open science work they're doing. Just quickly, I might have missed this, but what are the QC variables that you are outputting or you're planning to output?
The QC variables I'm trying to output are that the trial counts to see if there's any missingness, and the original validation. Sorry, not original. The output would be a markdown report with validation results, summary tables, relevant figures, and warnings. Like showing required column checks, trial checks, distribution, and yeah, I guess it's.
So it's mainly like task data. So about the task, about the behavioral data, and also the other thing is also the imaging data, right?
Yeah.
And how about resting state data, or are you just doing task data?
Oh.
That's a good
Let's take
suggestion.
the test
Yeah,
first.
yeah. I haven't. Right. Yeah, I haven't thought about the neural data set, but I would definitely I think I would also include resting state data, the neural imaging ones, other than just the technical, the behavioral ones. Yeah, I'll think about that.
Yeah. Okay.
And you can integrate it, I think, to this report to include. So for the behavioral part, you have all that, the RT, the missing trial, and right. But for the brain part, you can also include like how many of what type of images are they all there? Are they intact? Just by way of file validation.
Mm.
It's not even talking about the quality of the images in there. That's another thing. But do we even have the files? Are they there? Where are they? You know, do they make sense?
Oh yeah, yeah, that was okay.
Yeah. So that can be part of the report as well. And then of course the key thing is warnings, like to tell us where it's not making sense, and so that we can zoom in and solve those problems.
Okay.
Yeah. Thank you. All right. I think that's it. Okay, thanks, Chelsea. Let's move on to the next person.
Next person would be Jessica Lei.
Oh,
Okay.
Jessica, you're here.
Hi.
Where are you? Okay.
All right. Can you see my screen?
Yes, go ahead.
Okay. Hi, everyone. So I'm Jessica from NTU Taiwan. I come to this from the social linguistic background, which is partly why I'm interested in whether the language models can do the labeling work that human writers currently have to, especially working on a large data set. So my project tests whether a transformer can generate emotion labels that meaningfully agree with the human consensus on the naturalistic film fMRI datasets called EmoFilm. And okay, so here's the problem. Now, okay, so we have the rich naturalistic film fMRI datasets moment-to-moment brain responses while people watch real films. But to train a model to decode emotions from those responses, we need labels. So which emotions and when? And I think that's a bottleneck. So most of the datasets according to the previous studies, as you can see, cited here carry lack of emotions annotations and the standard fixed continuous hand annotations by human writers, which is very quite slow, costly, and doesn't scale. So can an automatic method close it? So lexical-based tools like Fader have been tried, and transformer classifiers like BERT have been barely tested for this. So I have two questions in this project. First, do transformer generated labels agree with human consensus? And second, this is the harder one for me, can they decode emotions from the brain as well as the human label do? So on the human side, my benchmark is 44 raters, 50 emotions items, and continuous consensus. So on the machine side, so we have BERT over the film transcript. So I start with valence 1x6 as the cleanest first test of agreement. And the data set is the EmoFilm open on open neural. And what makes it rare is built-in graph truth. So 14 short films, 30 participants scanned at 3 Tesla and emotions ratings from 44 annotators across 50 items. So the discrete emotions plus appraisals components grounded in the shaker of components process model. So that continuous human consensus is exactly what I validate the machine's label against. And the films are openly licensed, so I can recover the dialogue model's needs. And okay, so here the film, the film profiles. Briefly, this is just show a stimuli work. So these are the post-scan engagement ratings straight from the paper itself, table 1, absorption, enjoyment, and interest. And as you can see here in the heat map, so Sintel, right there, Sintel. high across the board, and at the bottom you again which is quite low, so the film span a real string. So to be clear though, these engagement score are not my emotion label, so they are from two places. The first one is the 50 item annotations already in the data set, and the birds labels I will work on later in my final project. So this slide is just an evidence the films elicit very responsive. And for the pipeline, so the pipeline have two parts. So the committed core now have three steps. One, recover each film. I'm gonna recover each film transcript. And the next step, I will try to run BERT for the sentiment score per dialogue segment. And the last one, the careful step, I think I will try to map that to a valence process and correlate it and second by second against the 44 rater consensus. I think this is a direct answer to where the machines agree with humans and where it fails. And then a stretch, I'm gonna take those label into the brain and decode valence from the bow signal, the brain activity signal with an SVM. And okay, so on standard and open tools. Okay, and I scope this, so there's always result. The committed part is kind of pure NLP and statistic generated labels, and then derive the valence process, quantify the agreements with the human consensus, and report whether it fails. So my core deliverable doesn't depend on the fMRI pre-processing. So that's referred for the stretch goal. So even if the brain data side is the problem, I think the validations result still lands on time. So decoding is upside, not a dependency. And why it matters? There are three reasons. So any decoder is only as good at labels, and knowing where automatic labels fails is still is still studying. And if they prove reliable, a large body of unlabeled data sets becomes usable. And it also extends the same method can move to cross-linguistic data, including some languages which is under study, like Vietnamese, which I'm aim for in the future research. So this measure emotions in film via language, not the language processing in isolation. So that's a natural follow-up beyond this project. Okay, so these are selected references. So in one line, I'm testing whether AI can label emotions well enough to trust, and building a method that scales beyond a single data set. And thank you.
Okay, thank you, Jessica. Any questions? Amanda has a question.
Yes, I
And
do have
then Pauline,
a few clarification questions. So the fMRI data set has like 14 films as the stimuli, and you transcribe the films into text. That's your plan work. Okay. And what are the 50 human labeled items?
The 50 human labels items?
Yeah, because you mentioned something like 50 human items as some kind of validation stuff.
Yeah, so actually that's in the paper itself. So there were 50 emotions labeled, but for my project, so I think like I cannot do that due to the limited time frame for the project. So I use the valence one axis, so as a tractable first test, not because the emotion is one dimensional, but because it's the cleanest place to ask whether the machine and humans agree at all. Yeah,
I was asking like 50 what? 50 sentences, 50 isolated words, because as we've discussed, BERT is really great at sentence level contextual embeddings, but not isolated words. So I was just kind of wanted to clarify that.
Okay, I got it. I will look into that again. Thank you.
Okay, and also have you tested the BERT classification model on like publicly available text data sets for sentiment analysis?
Not yet.
Okay. 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 You might want to do that first before
Yeah.
before the actual fMRI thing.
Yeah, definitely.
Okay.
Definitely. Thank
It's
you.
really interesting, and I think it's really ambitious. So really looking forward to your results.
Yeah. All right. Thank you.
Any comments? Yeah. Amanda, you had another question?
No,
Oh,
I was
okay,
clapping
right.
my hands.
No, I think, have you looked at the fMRI data yet?
Just a few, but because at this stage, I don't need to, touch the pre-processing,
I see. Okay,
fMRI data. So I just leave it later for the stretch goal for the,
Okay.
future work.
Yes, and I understand that, and this is fine. But I will also suggest maybe spend some time first to, do some planning, and go to at least one person's fMRI, and try to do some analysis, because I think, I think what you're doing is fine, but I feel like you will hit a kind of a barrier when you try to go to the fMRI, because you will find that the fMRI data does not fit the format that you want to do that testing. And so that even if you do all of that and you say it's independent for the fMRI, I mean, the big goal here is to touch some brain data. So you do want to kind of at the start already gear what you are doing, not so independently from the fMRI, but at least towards the direction that allows you a higher probability of integrating it with the brain data. So to do that, a critical thing is to first actually understand the fMRI data a little bit better, rather than to be so distant from it, because you will spend all this time doing all your thing, and then you find that none of it can be used for fMRI, and then then what's the, you kind of miss the goal and the opportunity here. So I would just think about that. Again, if you have questions, put it in the pod, make noise, and then it's the TA's job to help you with the fMRI and all that. Okay?
Okay, thank you.
All right. Okay, in interest of time, let's move on to the next person. Thank you so much, Jessica. Let's go to the next, who is next?
Next person will move to Singapore for Wen Liang.
Yeah, Wen Liang, are you okay?
Yes.
Can you share your screen?
Wait. Yes, let me see.
Can't hear you.
Testing.
Yeah, good. Okay. Can you share your screen? Okay.
And can you see?
Yes. We can see ourselves.
Yeah.
Okay, good. We can see a slide.
This one?
No, this one is your presenter view.
No.
Yeah.
You need to swap to the audience view.
Okay. This one?
Okay.
Yeah, looks good.
Okay.
Okay, go ahead.
So I'm Wen Liang. So today I'll be presenting understanding object recognition from naturalistic scenes. So I was interested in three-dimensional objects recognition, and then because of the computational constraints, I had to downscale it. So I chose naturalistic scenes. So this project is based on this paper. So the aim is to understand neurodynamics of visual object recognition using a large diverse set of natural images. Okay, so the two research questions that I aim to answer is firstly, to capture the differences in the neural activation patterns in response to diverse stimuli within individuals, and then also to capture inter-individual differences. Yeah, so for this project, I'm looking at the natural object dataset. So this is from the Open Neuro, and then so there are 60,000 images So how the participants watch it, so they view this image first, and then after that there's a fixation period, and then the next image comes up. So in total there are 19 participants. So there's multimodal data from this data set, but I'll focus on the EEG data. So 9 individuals viewed 4,000 unique images across 4 sessions, and then after that, the remaining 10 participants completed 2 EEG sessions, and they viewed 2,000 unique images. So in total, this whole data set comprises of 24 EEG data, 24 hours of EEG data with 56,000 images. Okay, so next, for the data collection, so the participants were asked to actively indicate whether the presented image was animate or inanimate, so whether it's animal or plant, or is it like a carbon or a structure. Then participants were involved, were also, so just something to take note, participants were involved in the experiment for all 3 modalities with the same stimuli. So there may be some sort of practice effects for EEG and MEG data which were collected later on. Okay, so right now I'm downloading the data. So for future directions, firstly that would be to analyze the accuracy and reaction times to these object categories. So whether they respond true or false to animate or inanimate objects. And then moving on, and then I will try to integrate the EEG data to the MRI data or MEG data, and then examine their activation patterns. So specifically looking at the ventral temporal cortex as the ROI, and then after that I will use a Spearman correlation to compare between the fMRI or MEG and EEG data. And then if time permits, then I'll move on to the brain decoding. So then I'll build a model to decode EEG signals and then try to recreate the images that the participants see. Okay, so thank you very much.
Okay, thank you, Anyang. So quick question. So have you tried the data yet? You said you're downloading.
Yes, I'm downloading.
But do you have like at least one data set yet? One person?
No, not yet. Still downloading.
Oh, okay. All right. So I would think that you might need to rescale your goals here. I don't know if you will have time or even like to do the work to really do that object recognition for fMRI, MEG, and then combine and all that. Okay. It might be more meaningful to just say like you want to choose one modality and do object recognition, the decoding or whatever it is, and even just for a few subjects. Okay, because based on experience, like this is not to do work with such a massive data plus multimodal is not trivial. So and we see this all the time, right? so I think at the start you should be aware. I think realistically you choose one modality, you choose a few subjects, and then you scale down the problem. Might be a thing more realistic within this time frame. Yeah. Any other questions?
Yeah, just to clarify, you're getting the data set from that study, is that right?
Yes.
Okay, so they have all 3 data sets over there.
So I downloaded the EEG data first.
Okay.
I'll prioritize the EEG data before I move on to the other modalities.
Yeah, I think then you just try out the EEG data to understand how it looks like. I think that's actually quite a lot already is. And also whether are you analyzing the raw data, right, from scratch versus that is already pre-processed. Because the pre-processing steps itself will be take quite a long time.
Yeah. Oh yeah. I decided to use the pre-process and the epoch data.
Oh, okay.
Yeah, in the interest of time.
Okay. Yeah, but yeah, just try and test it out first, see if it works.
Okay.
Okay, any other suggestions, comments? No? All right, then let's go on to the next person. You're
Next
almost done.
presenter is William. Hi, it's me.
Yes, William yourself. Go ahead.
You saw my screen.
Yeah, okay.
Yeah.
Don't think the mic on?
Wait, hello?
Okay,
Yeah?
yeah.
A little bit. Hi, this is William, and this is my topic, functional connectivity changes induced by a Lego robot.
You want to move that out of the way?
I should remove that, yeah. Induced by Lego robot inference training in aging brains. This is also my thesis topic. But since we are in here, Brain High School, so I will spend more time talking about methodology here. And this is my outline. I will talk about background, some review and method. The most important part is I compared with the Brain High School functional connectivity modules with my pipeline, and I encountered some difficulties, and I want to share with you. And to start with background, what is inference? You might think inference is basically just thinking, but actually deeper than that. We have to trace back to the theory behind it. Is the Carl Friston once introduced the concept of Bayesian brain and Bayesian inference. In short, our brain doesn't like uncertainty or unknown. So just like thermodynamic systems, our brain wants to minimal that kind of uncertainty, surprises, just like other systems. So how? By 3 steps, you receive a sensory input, so observe that is, and you change your belief or you form a belief inside, and you make a predictions and use the feedback of your predictions as another sensory input. So by that, you can minimal the prediction errors here. And why is inference so important? Because we believe that our brain is actually an inference machine. So basically, understand the mechanism of inference is understanding how our brain works. So so we recruit a group of 65-year-old above elderlies, and we conduct interventions. We recruit them to play some Lego robots and do some easy codings. But what is the Lego robot inference training different from other cognitive trainings? The biggest challenge of cognitive training is Lafarge transfer. That is, if I give you a course to writings, I'm good at writings now, but my memory or math still worse. But we're looking because we believe that inference is the basic of our brain works. So we believe that the inference training is likely to have far transfer. So by observe, by change belief, by actually predictions and actually conduct codings is adequate inference trainings. So the third concept is functional connectivities that I don't think I have to introduce too much. It's basically there are several approach to functional connectivities, including like our modules, ROIs to ROIs, functional connectivities, and my pipeline will be C to voxels connectivity, C to voxels correlations. And this is my aiming. Back to our topic, functional connectivity changes induced by inference trainings. So of course it will be a support to ICT project, but most important part is we want to know what is the critical connectome which achieve our progress in our trainings. Spoiler alert, there is a far transfer and behavioral data still improve after the interventions. So this is my method. Sorry. Compared to task-related functional connectivity, task-related fMRI analyze, there are several differences. First, it will need a stricter denoise, and second, we believe that smoothing and normalizing do contaminate correlations. This figure is this figure is provided by Xinqi, which we have a discussion a lot with. As you can see, if you start your correlations analyze with smoothing and normalizing, on the left, the figure on the left, there are more strong correlations appeared, which they are artificial and we don't want. They are not intrinsic synchronization we want to receive. So what happened? Oh yeah. So of course, without normalization, we can use Atlas. Of course, we can do comparison between subjects. What do we do? So this is my pipeline. So of course, in the beginning, we have a seed ROIs in MI space, of course. But we can do correlation analyze in MI space. So by the deformations field, we can transform it into subjects, the subject specific seeds, and we do correlations in subject space, and inverse transform it into back to 3D image. Then we can transform back to MI space and ready to second level analysis. So sorry, because of time, I will skip a little bit. So this is the C we will choose by our senior Ziyun. We choose in the brain head school project. I choose hippocampus, right hippocampus, and left parahippocampal gyrus, because we believe that they are responsible for forming belief in inference. So a brief contrast to brain head school pipelines. When I put my data inside their pipelines, the matrix come out live good. Then this come out. Of course, as you can see, they are not correct alike, because their data to start with is smooth and normalized. So a little suggestion to how to do is we can make a subject specific AL atlas using deformations, and you can continue your brain head school pipelines to ROIs correlations. These are all also made by me. So thank you, everybody, and allows me to save my result for upcoming Singapore trips. Thank you.
Okay, thank you. Very exciting work. Any suggestions or questions for William? No. Okay.
I have a question.
Go ahead.
Hi.
I'm just curious. Like, do you have a? Can you go back to the slide where you showed two correlation matrices? One with smoothing and normalization, the other without.
1 sec. This one, or
sorry,
this one.
The previous one, I think.
This one, yeah.
Yeah. Have you done like one with smoothing but without normalizing, and one with normalization but without smoothing? Because like smoothing definitely increases correlation.
Yeah.
Because basically, when you smooth the voxels, the voxel time series becomes like essentially linear combinations
Yes,
of
they were very
neighboring voxels. But I am not really sure about normalization, like mathematically speaking. So I'm just curious if you've done these two, preprocessing steps independently.
Oh, to be honest, no, we haven't. Sorry, 1 sec. No, we haven't tried just smoothings or normalizations.
Yeah, so maybe it's just the smoothing, not normalizing. I'm not sure. Just mathematically speaking, yeah.
But I believe that deformated together those voxels are deformated together. Maybe some kind affect the correlations between them. But of course, we can try that out in codes, of course.
Okay, cool. I was just curious. Thank you.
Thank you.
Okay, great. In that, if no further things, then let's move on quickly to the next, second last person.
Okay, the next presenter is
Who is
Fan Yukang.
Yu Kan. Thank you for holding out. We are severely overdue, but thank you guys for sticking to this.
Hello.
Yes,
Can
go
you
ahead. Are you able to share your video, your webcam?
Camera, okay.
Yeah, thank you.
Okay. Hi everyone. I'm Fan Yukang from NTU Taiwan, the Department of Psychology, and today I will present my research project on dynamic functional connectivity of the brain during sensory deprivation. Let me briefly introduce my idea. In my engineering background, I initially see the brain as a machine that relies on constant external sensory input to calibrate its reality. And I want to use the sensory deprivation method to test what exactly happens when these external stimulus, like our visual stimulus and hearing, are suddenly lost. My goal is to investigate deprivation-induced phenomena and assess the underlying neural mechanisms. According to predictive coding theory, the brain functions as a prediction machine. It constantly balances high-level top-down prediction against bottom-up sensory input. But what happens when the brain faces data starvation? That is my previous question that I want to figure out. Yeah, to explore this, I use a method called sensory deprivation, which means eliminating as much input as possible, aiming for total darkness and absolute silence. And the goal is to completely block out the visual and auditory inputs, the two major channels that humans use to anchor ourselves to reality. So by throwing the brain into total darkness and absolute silence, we force it to operate without bottom-up calibration. Looking at the past research, we see a critical trade-off between deprivation completeness and duration. Like for example, in 2009, the Mason study achieved extreme silence under 10 decibels and induced anomalous effect like hallucination in just 15 minutes. And in contrast, the previous study, like the Zubek in 1974, used like 30 to 40 decibel quiet room and needed many days. So the rule is quite simple that the higher thoroughness of deprivation leads to a faster onset of abnormal states like hallucination. And also there's other variables like individual hallucination proneness also heavily influence the results, but I will just go through it. Yeah, so because of that, we need a highly controlled environment. I compared two conditions. The high degree sensory deprivation use a professional and anechoic chamber, which can make a strict environment of less than 10 decibels and total darkness. But however, we can't do real-time brain scanning like fMRI or MEG in this kind of environment. So for this class project, I choose the low degree sensory deprivation that using the air plugs and eye mask to simulate the deprivation state and do the MEG scanning. Yeah. So when the bottom-up signal is silenced, previous studies often focus heavily on behavioral hallucination task. But for my project, however, I will drop the complex behavioral task and focus primarily on neuroimaging through MEG scanning. And okay. So here's the single scanning flow. The total length is approximately 15 minutes. And it starts with a 10 minutes pre-resting state, but I will do it without fixation point. Yeah. Then the 30-minute main session begins, where subject maintain the sensory deprived state wearing an eye mask and earplugs while executing a press and hold button task to monitor wakefulness. And finally, we record a 10 minutes post-resting state baseline. Afterward, we conduct an interview to verify if they experience any abnormal feedback, like reporting hallucination or other state. Yeah. So the core measurement tool for this project is dynamic functional connectivity. Yeah, my focus is on tracking network variability over time, not the traditional functional connectivity analysis. This is the core pipeline I will use. I will go through it briefly. First, we extract the time courses from all brain regions. Then the second, instead of averaging the whole 30 minutes to do functional connectivity, I'll use the sliding window method to generate a continuous series of connectivity metrics. And third, by applying clustering algorithm, I can identify specific microstates the brain enters. And the ultimate goal is to extract dynamic features like real time and transition probability to objectively quantify how data starvation destabilize the brain system at a network level. Okay, the final thing is to briefly touch upon where this project currently stands. My data is entirely self-collected primary data conducted in collaboration with Professor Brown-Shear's Brain Consciousness Lab. And the administrative process for the IRB and MEG Center application are still under review. Maybe due to the risk of this kind of research, need more time to check. And yeah, so I haven't acquired the actual subject data yet. But once I get the administrative green light and complete our dry run, the dynamic functional connectivity analysis pipeline will be ready to process this real-time scanning data immediately. Thank you for your time.
Okay, thanks. So as I know with IRB, 不乐观. So
I'm not sure about it.
When did you all start? When did you all start applying for IRB?
1 month ago.
I mean, you know, it depends on how they think about it. But anyway, what if you don't get the IRB and you cannot collect the data in time? What are you going to do within the Brain Hack School? What's your plan?
I think I will collect open data of resting state to do the same analysis of dynamic functional connectivity.
Actually, I was wondering if you wanted to do sleep scans, which is, I mean, they're sleeping, but they are not, they're internally, they are shut off from the sensory stimulation.
Oh, you see, do you mean let the subject go to the MEG scanning and they sleep?
Yeah, they sleep, or even in fMRI, it doesn't matter in this case.
Maybe if they reject me, I will change my proposal.
Oh, I see. Well, but I'm saying that sleep data in open science, there should be people sleeping, I'm sure.
Oh, okay.
Or you just tell Brown to let you put someone in there and sleep.
Yeah, I think he will agree.
Don't need ARB, put yourself inside, sleep, collect the data, and then you're done. Yeah. Right. But anyway, okay, so some suggestions about, since you are stuck at the data thing, it's very interesting. And if you cannot collect the data, these are some suggestions to get around that. Okay. Yeah. Any questions, suggestions? Yeah. Kevin?
Yeah, just a minor suggestion. So since you are using like dynamic functional connectivity measures, I think you might consider using like instantaneous frequency to get the features, because even with sliding window, you still lose the difference between time, because you are looking at like this window by window base. But instantaneous frequency will just give you at least technically they are using like 3 time point or 5 time point to identify instantaneous phase. So it will be like more dynamic features there. Yeah, just one suggestion. Yeah, and you can do that in using like Hilbert transform. Is also implemented in ML Python. Yeah.
Okay, thanks for
some suggestions for the features to look at. Yeah, it's always a problem when you're looking for dynamic stuff. It's very hard to define the window and all that. So,
just echoing what Josh mentioned about safety for your data. You only have 2 weeks left. I would suggest, because you're learning actually the dynamic connectivity function, connectivity method, right? And understanding what Kevin already provided some input. Perhaps you just get some open source, even just resting state data. It doesn't answer your question that you're interested in, but at least you built up the experience of that, the pipeline for analysis at least works, right? Something is working, and that you can later apply to your study if it's approved by the IRB. So
Yeah.
that would be more feasible within 2 weeks.
Yeah, I think you might seriously have to think about that. Like either you get open data of MEG resting, right? I think basically don't wait for IRB, because that is something you cannot control, and the longer you wait, the harder it is for you to actually validate, right? So, but what's guaranteed right now is if you go online, you probably can download immediately, like today or tomorrow, resting state MEG data. That's one. Another one I was going to suggest was, as I said, put yourself in there. Then no one can say anything about the IRB. I wanted to sit in there and collect my own data. What's the problem, right? And then analyze your own data. You yourself sit there and rest an MEG. If you already had the auditory sensory deprivation set up, do it to yourself. Collect your own data, analyze your own data, just to familiarize with the technique. That's one thing that you could do. I'm sure the MRI Center, as I know, would be okay with that. You can tell Brown and ask Brown to talk to the center, and I think they for lab technical testing, you should be okay.
Okay.
Yeah. Okay. So some suggestions to keep you going instead of waiting for IRB. Yeah. Okay. Thank you so much. Let's go to the last person.
Okay. Our last presenter is Li Nan. Li Nan
from Singapore, is it?
Oh, hi.
Okay, great.
Okay, so hi everyone, my name is Ling Nan, and my project is called Age-Related Difference in...
Can you turn on your video, your webcam, so that...
Oh, sorry.
If it's okay, yeah.
Okay, hi. So hi everyone,
Yeah.
my name is Ling Nan, and then my project is called Age-Related Differences in Residing States Foundational Connectivity Using Open fMRI data. So my idea was more focused on organizing and visualizing on open neural data set. For my final project should directly work with neuroimaging data in a more meaningful research sense. I plan to work directly with residing states fMRI data, extra foundational connectivity feature, and compare younger and older adults. For background, why connectivity, aging is often associated with change in cognition and brain function. For example, older adults may show differences in memories, attention, processing speed, and other cognitive abilities. Residing states fMRI is useful for studying aging because it measures spontaneous brain activity when participants are not performing a specific task. From residing states fMRI, we can isolated foundational connectivity, which describe how activity in different brain region is relative over time. By comparing functional connectivity between younger and older adults, this project can be explore age-related differences in brain network organization. So for my research question and project aims, my research question is how does residing states functional connectivity difference between younger and older adults is an open neurocognitive aging fMRI data set. The goal is to move beyond basic data set organization and perform a small but concrete neuroimaging analysis. Specifically, I want to extra ROI level time 0 from the fMRI data, compute functional conductivity matrices compare younger and older adults' connectivity pattern and report the limitation clearly so the projects stay realities. So for dataset and tools, so for the dataset, I plan to use OpenNeuro DS003592, which is a neurocognitive aging dataset. It includes 301 healthy adults, 181 younger adults aged 18 to 34, and 120 older adults aged 16 to 89. So the data set include behavioral measure structure, MRI, and 2 10 minutes multi-echo resting states fMRI runs. For tools, I plan to use Python and Jupyter Notebook for the analyze. I may use libraries such as Pandas and NumPy for data organization, Nylern for fMRI, and Atlas-based analyze, and Matplotlib or Seaborn for visualization. I will also use GitHub to organize the code and documentation and to create a simple web-based project page for final project. The planned workflow was several steps. So first, I will select manageable subset of younger and older adults so that the project remains feasible as an individual project. So second, I will load the resident states fMRI data and check whether usable derivative or per-processing output are available. Third, I will use bring Atlas to extra ROI level time series from the fMRI data. First, I will compute foundational connectivity matrices by calculating correlation between ROI time series. Finally, I will compare connectivity pattern between younger and older adults using visualization and simple statistical analysis. The mean output will include connectivity heat map, group comparison, and comparison plots, and probably select network level summaries. So the exact delivery of deliverable are structured GitHub repository and Jupyter notebook showing the analyzed workflow, foundational connectivity matrices, group level visualization, basic statistical comparison, and a simple web-based project page. I will start with a small balanced subset of participants and focus on Atlas-based foundational connectivity analysis rather than a full large-scale fMRI processing pipeline. I may also explore whether behavioral measure different between age group or related to connectivity feature. Overall, this project directly worked with neuro imaging data, includes feature extraction, visualization, statistical comparison, which makes more appropriate as a final project. So these are the main source supporting the data set. Yeah, that's all.
Hey, great. Thank you so much. Very good. Yeah, sounds very practical, very doable. And it sounds like you know that you probably can't do it all, so just do some. So that's very good. Any questions or recommendations?
Yeah, just a comment, Linan. So how many, when you say manageable set, right? Because you probably haven't done any of this before, so it's like really testing out. What is the manageable numbers you have in mind?
maybe fuel. Maybe I'm not sure. Maybe
Perhaps you can run like you're thinking of doing the pre-processing everything, right? So maybe just try one subject first to run through the whole pipeline.
Okay.
If it works for that, and then if you're comfortable with that, and you learn, go into the fMRI pod or the MRI pod, then you learn about scripting and all that. Then you can do a whole batch, and that will be faster.
Okay.
But it really depends on how, if you're doing it locally, how fast your computer is and all that stuff. Yeah, but definitely at least try one first to get a feel of what it means, or what are the factors or variables you need to pay attention to.
Okay. Yeah.
Yeah, I would suggest if you try one and you're comfortable, then try try a young adult one, right? Then if you're comfortable, try an older adult one.
Okay.
And then you get 2 heat map, then you can at least do some also another kind of almost group level kind of thing, even if just very simple. That if you can, if not, just stick to one, it's fine. Yeah.
Okay.
Yeah.
Thank you.
And just a question about the original data set that you picked from. What was the original purpose of the data they collected? Like what are they comparing between the age groups?
Let me see. 301 healthy adults, 181 younger adults, age 18 to 34, and 120 older adults, age 16 to 89.
What was the original purpose of them having that data collected? Did they mention in the data set? Or because I asked this question is just maybe first of all, most importantly, just to get comfortable with how the whole pipeline and processing work is done. But also, I think to add a little bit of also a nice theoretical look at it is probably just, is this pipeline dimension or something reproducible, or like what's the comparison that's targeted in this sense? Is it more like some people say more efficient or different? That sort of thing.
Okay.
I think you can read.
I need to
read their original paper more. But if you can replicate, but replication means you must do the whole thing, right? But if you can, it's okay.
Yeah. Okay.
But at least you understand where the data are coming from, and I think that's also a pretty important thing. Yeah.
Okay. Thank you.
Okay. It's awesome. We look forward to what you can deliver, and I think that's it. All right, yay!
Yeah.
Great job, everybody. Give yourself a pat on the back for those who stick to the end, and for those who are online and stuff. It's really a marathon. But I think this year we have quite a lot of interesting and novel projects as well compared to last year as well, right? So I think all the NTU teachers will agree with me as well. And I think we very much look forward to your final project presentations. And then I think the next session is next Monday. Is it the industry talk on Monday? Okay, Monday is Zhongyang that side will be hosting their industry session, and we'll be posting. I think we've posted the information online. Yeah, but we'll remind everyone again. Otherwise, we'll see you guys online if you can, in the industry session. Okay.
Yeah.
TAs have any other announcement, or Singapore
Next
Science?
Monday is, I think, Vesak Day holiday for right.
Is it?
But nevertheless, the students do put on the post